In this video, we'll learn how distant cells communicate with each other. Activities that are carried out inside cells, tissues, and organs occur in different parts of a multicellular organism and are coordinated by long-distance signals. These signals are usually small molecules that may be transported through plant tissues within organizations such as the symplast, meaning shared cytoplasm, or apoplast, meaning travel through extracellular space. As shown in this image, signaling molecules can pass through symplasts, which are connections between adjacent plant cells. These connections are made possible by structures called plasmodesmata, which interconnect cells to form a continuous membrane system, the symplast. So the cell membranes of adjacent plant cells are continuous with each other, resulting in a shared cytoplasm between adjacent cells. The other route that signaling molecules can take to travel through the plant body is via the apoplast, where molecules move through the extracellular space that exists around plant cells. The molecules that carry messages from one part of the plant or animal body to the other are called hormones. A hormone is an information-carrying molecule that is secreted from a cell. It circulates throughout the body and acts on target cells that are far from the signaling cell. Hormones are usually small molecules, are typically present in minute concentrations, and have a large impact on the condition of the organism as a whole. The chemical structure and function of hormones may vary widely. They may be soluble or insoluble in lipids. Hormones that are soluble in lipids usually diffuse across the plasma membrane easily, since the plasma membrane consists of a lipid bilayer. These hormones then go into their target cell's cytoplasm to communicate the signal to the interior of the cell. Lipid-insoluble hormones, on the other hand, are large or hydrophilic, making them unable to cross the plasma membrane. Instead, they bind to a receptor on the cell's plasma membrane, which allows them to transmit the signal to the interior of the cell. Some examples of hormones are included in this table. We've got some plant hormones that include auxin, prostanosteroids, ethylene, and cysteamine. These hormones mainly affect plant growth and development. Examples of animal hormones include estrogens that play a very vital role in development in animals, FSH, which, like estrogens, is involved in the reproductive cycle in animals, and then there's insulin, which regulates glucose levels in the body, prostaglandins that serve multiple different functions, and thyroxine, which regulates metabolism in animals. Let's look at how steroid hormones carry signals into receiving cells. Steroid hormones, being lipid-soluble, bind their receptor on the plasma membrane of the receiving cell. Once the steroid hormone binds its receptor on the target cell, the complex then travels inside the cell and reaches the nucleus. It goes through the nuclear pore and can then initiate transcription of the target genes. The activated gene is then transcribed by RNA polymerase. The resulting RNA exits the nucleus through the nuclear pore and is translated into the protein that allows the cell to launch a response to the signal. Let's look more closely at the steps that are involved in cell-to-cell signaling. This process occurs in 4 steps: signal reception, signal processing, signal response, and signal deactivation. During signal reception, molecules called receptors that are present on the plasma membrane of the receiving cell or the target cell are bound by hormones or other cell-to-cell signals. The presence of an appropriate receptor protein dictates which cells will be able to respond to a particular hormone. There can be identical receptors in diverse cells and tissues that allow long-distance signals to coordinate the activities of cells throughout a multicellular organism. Signal receptors are proteins that change their shape or activity after binding to a signaling molecule. This change allows the signal to be transmitted to the inside of the target cell. Signal receptors are dynamic. They may change their sensitivity to particular hormones, and their activity can be blocked. This property is often utilized in the pharmaceutical industry. Signal receptors that bind to lipid-soluble hormones are located inside the cell, since these hormones can diffuse through the plasma membrane without any problem. Other signal receptors are located in the plasma membrane and bind to hormones that cannot cross the plasma membrane. Lipid-soluble hormones bring about a different response in the target cell than lipid-insoluble hormones. Lipid-soluble steroid hormones bind to receptors inside the cell, as we saw in the simulation earlier, and trigger a change in the cell's activity directly. The hormone receptor complex is transported to the nucleus, where it alters gene expression. So when a lipid-soluble signal, such as a steroid hormone, travels inside a target cell, it binds to its receptor in the cytoplasm, resulting in the formation of a hormone receptor complex that then travels into the nucleus and alters gene expression. Activation of target genes results in the formation of protein products that bring about a response to the signal. Lipid-insoluble hormones that cannot diffuse across the plasma membrane bind to receptors that are present on the cell surface of the target cell, which triggers a complex series of events collectively called a signal transduction pathway. This converts the extracellular hormone signal to an intracellular signal. The message transmitted by a hormone is amplified as it changes form. Transduction of the signal occurs at the plasma membrane, whereas amplification occurs inside, meaning that even if the signal is present in very low concentrations, once it binds to the receptor on the plasma membrane of the target cell, that small signal is amplified on the inside of the target cell. So to recap, when the lipid-insoluble extracellular signaling molecule binds its receptor on the plasma membrane of the receiving cell, there is a change in the receptor that allows the transduction and amplification of the signal inside the cell. This amplified signal then reaches the nucleus and alters gene transcription. The activated target genes then produce protein products that serve as a response to the incoming signal. Let's view a simulation of the process. When an incoming signal binds its receptor present on the cell surface of the target cell, it brings about a change in that receptor that leads to the activation of a previously inactive enzyme that binds and processes its substrate to generate multiple second messengers that carry the signal and amplify it on the inside of the cell by binding to and activating other target enzymes, which generate further protein molecules that are called effector molecules, that then activate other enzymes that bring about a response to the signal. As we can see, the signal is also amplified through this process. Signal transduction involves multiple proteins. Examples of these proteins are G proteins or enzyme-linked receptors. G proteins trigger the production of an intracellular messenger, whereas enzyme-linked receptors trigger the activation of a series of proteins inside the cell. G proteins are intracellular peripheral membrane proteins that are closely associated with transmembrane signal receptors. When G proteins are activated by a signal receptor, they trigger the production of messengers inside the cell. They link the receipt of an extracellular signal to the production of an intracellular signal They are activated when they bind GTP and are deactivated when they hydrolyze the bound GTP to GDP. This allows controlled activation of the G proteins. So how do G proteins transduce signals to the inside of a cell? When an external signal binds receptors called G protein coupled receptors, a change in these receptors activates the G protein, which is initially in the off conformation and is bound to GDP. When the G protein becomes activated, it exchanges GDP for GTP and splits into different subunits, one of which binds to an enzyme located further along within the cell membrane that then results in the formation of a second messenger that carries the signal further in the cell. These second messengers are small molecules that diffuse rapidly throughout the cell and amplify the hormone signal. Several second messengers work by activating protein kinases, which are enzymes that add a phosphate group to other proteins. The process of adding a phosphate group is known as phosphorylation. So kinases phosphorylate other proteins when they are active. Different second messengers bring about different responses in the target cell. Some examples include cyclic GMP, which opens ion channels and activates certain protein kinases, DAG, which activates certain protein kinases, IP3, which opens calcium channels to transport stored calcium ions, cyclic AMP, which is a very common second messenger inside cells that activates certain protein kinases, and calcium ions that bind to a receptor called calmodulin to form a complex that then activates other proteins. Other types of receptors include enzyme-linked receptors, which are transmembrane proteins that bind a hormone signal and directly catalyze a reaction inside the cell. The best-known group of enzyme-linked receptors are the receptor tyrosine kinases, or RTKs. So how do RTKs transduce signals to the inside of the target cell? They do this by catalyzing an intracellular reaction that involves 5 steps. First, the hormone binds to an RTK, after which the RTK forms a dimer and is phosphorylated by ATP. Next, proteins build a bridge between RTK and the RAS protein is subsequently activated. RAS is one of many proteins that are involved in the transduction of the signal. RAS then triggers the phosphorylation and activation of another protein. This process or phosphorylation cascade amplifies the original signal many times over. The cascade involves the repeated process of each phosphorylated protein catalyzing the phosphorylation of subsequent proteins. We can see the RTKs at work in this image. The external signaling molecule binds and activates RTKs, which then dimerize, meaning 2 subunits of the protein form a complex to become activated. Once activated, the RTK catalyzes phosphorylation of itself. This phosphorylation allows the RTK to bind to bridge proteins that connect it to a protein called RAS. When RAS is activated, it brings about phosphorylation of other kinases inside the cell, which triggers a phosphorylation cascade, leading to a particular response to the incoming signal. Many of the key signal transduction events observed in cells occur via G proteins or enzyme-linked receptors. These events have 2 results. 1, it leads to the easy transmission of an extracellular message, which is converted into an intracellular message. And the original message is also often amplified many times over. So you need only a small amount of signal to trigger an amplified response to the signal. How do cells respond to incoming signals? The ultimate response to a cell-to-cell signal varies from signal to signal and from cell to cell. So it just really depends on the signal and the properties of the target cell. The response falls into two general categories. The first is a change in which genes are being expressed in the target cell. And the second is the activation or deactivation of a particular target protein that already exists in the cell. The last step in cell-to-cell signaling involves deactivating the signal. Turning off cell signals is just as important as turning them on, because if a signal is continuously on, the cell will keep responding to it, even when the response is no longer needed, and that can be harmful to the health of the cell and the organism. Cells have automatic and rapid mechanisms for signal deactivation. These mechanisms allow the cell to remain sensitive to small changes in the concentration of hormones and the number and activity of signal receptors. The end result of cell sensitivity to hormone signaling is an integrated whole organism response to changing conditions that occurs both inside and outside the multicellular organism. In addition to receiving a particular signal, there can also be interactions between signaling pathways. Each cell has many intracellular and membrane-bound signal receptors, which allows it to receive an almost constant stream of different chemical signals about changes in the environment. The signal transduction pathways that are triggered by these signals and receptors intersect and connect to form a complex network that allows cells an integrated response to an array of extracellular signals. The cell launches an integrated response as a result of crosstalk between these different signaling pathways that are activated by these diverse signals in the receiving cell. Elements or products from one pathway may affect another pathway, ultimately affecting the overall cellular response, which can be reduced when one pathway inhibits another or can be increased when one pathway stimulates another. At multiple points, the cell can regulate the flow of information. This allows the cell to respond appropriately to many simultaneous signals. This makes sense because we're constantly receiving not just one, but multiple stimuli from our environment that result in the production of multiple signals inside the body that can all bind to their respective receptors on a single cell surface and activate different signaling pathways that connect with each other, and this crosstalk results in a launching of an integrated response to the various incoming signals. We have reached the end of the video on distant cell communication. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.